Welcome, everyone. Thank you for joining us with our Integrated Pest Management training for the Pollinator Habitat Grant. Slide. So we just started off with introductions. We wanted to get into a presentation from the Ventura County RCD staff members on IPM, kind of introducing IPM, and then roll into Sarah Cusser from the Santa Barbara Botanic Garden, talking about pollinator monitoring, and then we'll go into Q&As at the end. This training is required by CDFA for those participating in our Pollinator Habitat Program. The goal is, or the hope is that all this pollinator habitat that you're implementing through the program will be accompanied by IPM practices. We want to, so RCD is going to start off with reviewing methods of biological, cultural, physical, and chemical controls to manage pests. And we hope you stick around until the end and we get to learn from each other today. All right, so a little bit of background on CDFA's Pollinator Habitat Program. This is the first year that the grant has been in place, and their goal is to address the urgent needs for pollinator habitat across ag land in California. So this grant prioritizes the planning and establishment of native habitats using locally appropriate native plant seed, where possible. You know, we all know that pollinators, bees, butterflies, and other species, so many other species, play a critical role in our productivity for our ag lands, but their populations are in decline due to various factors, including habitat destruction, pesticide use, disease, climate change. So we're grateful that all of you have chosen to participate in this program to help address this need. CDFA awarded organizations throughout the state of California this funding to so that we are the pass-through to farmers and ranchers to install this pollinator habitat and implement management practices that support pollinators. I think I'll go to the next slide. So the organization that was awarded this grant was Kachuma RCD, Kachuma Resource Conservation District. They're based in Santa Barbara County. And they're the financial manager, financial pass-through. All contracts are through them. We wanted to kind of break down the 4 main organizations involved in this grant, just so that it's clear our various roles. Community Environmental Council is a contracted partner by Kachuma RCD, and CEC is helping farmers with their applications and technical assistance, all the way to helping with filling out the appropriate reimbursement documentation so that they send the right documents to Kachuma RCD. Ventura County RCD, we're contracted by Kachuma RCD, and we are offering technical assistance and this IPM training. And Santa Barbara Botanic Garden is also a contractor of Kachuma RCD, and they're really the plant specialists of this group and are helping so much with selecting plants as well as with monitoring pollinators. All right, without any more delay, let's get started with John and Christina's presentation on IPM.
So I'm going to start the presentation on IPM, and I'm going to go more about a background approach to it, and then Christina's going to go on halfway through to talk more about the management practices through IPM. But so I'm going to start. Next slide, please. So what is IPM? And IPM is a sustainable approach to pest management that combines various methods, including biological, cultural, physical, and chemical control methods, ultimately used to minimize economic, health, and environmental impacts. Now, why does that matter? And the first reason is going to be because of the farmer's economic benefits to using IPM methods. And the first one is going to be by lowering your input costs on the farm, whether that ranges from fertilizers, pesticides, soil amendments, you name it. It does take a while to actually see cost savings on that, but once you actually implement more of these IPM management practices, you actually start saving costs compared to actually using fertilizers and pesticides and stuff like that. Next, there's the approach of environmental sustainability and impact, and that's by supporting biodiversity by protecting beneficial insects and organisms, all part of a healthy ecosystem, as well as using less chemicals that ultimately reduces runoff into the groundwater and improving our soils also. And then there's a reduction in pesticide resistance. So obviously, if we're using a lot less chemicals, you're preserving the effectiveness of these pesticides and herbicides for the day that you do need to use them, they're actually going to work. Next slide. And before I go on about how to approach IPM, I do want to mention that when we do use chemicals, always look, there is a bunch of warnings on that pesticide label on how to protect humans and stuff like that. But a lot of pesticide labels or herbicide labels also include warnings for environmental hazards and pollinator warnings. And this is a copy from a insecticide, and it just tells you how to protect the pollinators. So it describes how you should apply this insecticide after sunset, to apply when the temperatures are below 55 degrees, notifying your local beekeepers, etc. There's just keep an eye out for these labels and these warnings. So approaching IPM, and the way I like to approach IPM is with a circular diagram. And in this case, it's prevention, monitoring, thresholds, and control. And truthfully, there's no right place to start in this diagram, except thresholds. You would never want to start in thresholds, but I'll get into that a little bit later. But for this case, we're going to start with prevention. Next slide. And so there's a lot of different things farmers can do to help prevent diseases and pests on the farm. And the first one we can start off with is site location. So that's going to be testing the ground, the soil, looking to see what's growing on that land, making sure it's feasible for a new farm, or if you're ripping out an old farm or you're purchasing new land, just making sure your farm is going to work out like efficiently there. There's nothing in the ground. Then there's crop rotation if it's applicable to you. Typically, it's mainly more in row crop systems, but you want to rotate your crops so you can break life cycles of diseases and pests. And then there's also planting resistant varieties, whether that's a rootstock or a scion. They make a bunch of new varieties now of rootstocks and scions. So if you can find one resistant to something that you tested for on your field, try to pick that if you're growing a new orchard. And then there's sanitation also, and that's like in the photo right there, cleaning tractors, cleaning tractor wheels, cleaning your tools. I mean, in extreme cases, I've seen farmers that make you clean your boots before you enter their farm. So it's just making sure you're not transferring pests and diseases and soil-borne diseases onto different farms. And then there's proper irrigation and drainage. A lot of diseases start from over-irrigating. So as long as you can implement a healthy, efficient irrigation system, that helps a lot in preventing a lot of diseases and pests. Next slide. Now, there's the steps of monitoring, and monitoring is a crucial step to help understand what pests and problems exist on the farm and knowing when to act. And truthfully, it's really hard to monitor sometimes without knowing what's out there. And so there's these little booklets down here on the bottom of the page, and they're created by UCANR, the University of California. And each booklet has different components. So there's a booklet on all the weeds in California. But not only does it describe all the weeds and show you what they look like, they also tell you how to treat them, whether you use pre-emergencies or use what time period to spray them. There's also booklets on the types of pests you'll find in vineyards, the type of pests you'll find in landscapes, and the types that you'll find in orchard systems. And same concept, it'll show you what they look like from a young age to an older age, and just how to treat them and how to take care of them. It also shows you the type of damage they do on plants. And the best way to monitor for insects is going to be honestly with yellow sticky traps. And using pheromone traps, sweeping nets, and then from there, you're able to catch these pests. I recommend using the booklet to identify what it is. That way you can reach out to your local PCA or CCA to help them tell you what pesticides to use. And then for weeds, the best way to monitor for weeds is going to be keeping a germination log. That way you can log all the times that the different weeds grow throughout the years. That way you know in the future what IPM methods to implement so you can control these weeds, or if you need to use herbicides, you know when to use them, or pre-emergencies. And then monitoring for diseases, that's a bit tough. Usually I say if you see any area in your farm where the plants are stressed or they look like they're growing something weird, take a tissue sample, get a sample of soil, submit that to the lab, and hopefully from there, they're able to help you determine what it is and how to actually approach that. Next slide. And now we're going to talk about thresholds. And I can see the mouse really quickly. So the first threshold definition we're going to talk about is the economic injury level, and that's this red line right here. And so basically, the economic injury level is when the cost of pest control equals the savings from the crop damage avoided. It's also known as your break-even point. So this squiggly black line is going to be our season, or it's going to be our season. So this is your farm as a season progresses. Then there's this red line right here, pest population density. Density is low here, density is high here. So we can see that during this season, our pest population is going up and down. But what we don't want is for it to pass this red line here. Once we pass that red line, it's costing you more to farm than you're going to be making. So the way we go about this is we set up a economic threshold indicated by this blue line right here. And that is when the pest density is at a point which control action must be taken to avoid economic loss. So basically here, when we reach this blue line right here in this area right here, that is when we should take action. And so here you can see that they kind of took action, it worked, whatever they did, and it goes back down. And then here we see that, it's surpassing that line. And these dashed lines right here, this is the time frame that you have to actually take action of the problem, whether it's a pest, a disease, before it gets out of hand and it surpasses the red line, the economic injury level. And to determine the time frame between these dashed lines, it can be a bit complicated and it can cost a bit of money, but typically you'd have to reach out to a certified crop advisor, a pest control advisor, or sometimes the UC extension advisors sometimes have some money to help farmers determine these thresholds. But basically, this is what a threshold is. And that is my portion of the presentation. Does anyone have a question for me by any chance?
Okay, so we're now going to move into the control methods of IPM. So I know that you are all experts of IPM and pest management on your farms. So this is kind of just a refresher and to get us in the headspace so we can kind of talk about it after this. So IPM, the control methods, we think of as a pyramid. So the base of our pyramid is kind of the bulk actions that we want to be taking to follow the IPM protocols. And then the top of the pyramid, the smallest part, that's kind of the smallest amount of the protocol and the smallest amount of the actions we're taking on our farm when a problem comes up. So we want to start with cultural controls. And then when that doesn't work, we move into physical mechanical controls. And then we can go into biological controls, which starts to introduce more biotic life into our ecosystem. And then our last resort is chemicals, which include insecticides, fungicides, and herbicides. And yeah, we use it as a last resort, but it's still included in IPM because, you know, sometimes pests require different protocols to eradicate them. So to begin with cultural controls, which is again the first control we should think about when an issue arises. So these controls are practices that reduce pest establishment, reproduction, dispersal, and survival. So this kind of is making sure our environment isn't suitable to begin with for these pests that may come about in our crops. So examples of these controls are changing irrigation practices to reduce pest problems. So you can contact your local RCD to get an irrigation evaluation, or you can look at your irrigation to see if it's, you know, watering some areas too much, or, you know, if there's a problem there. Also measuring and managing soil pH. So again, kind of nipping the problem before it becomes, you know, kind of takes over the farm. Soil sterilization, hand weeding, removing debris and infested plant material, and crop rotation are all good examples of cultural controls. So if we tried some of those and we changed, you know, our environment to make it unsuitable for pests, and we're still seeing the pests on the farm, we then move into the second step in the pyramid, which is physical and mechanical controls. So these are tangible items that we're putting onto the land to get rid of pests. So this could include killing a pest directly, like with a rodent trap, like gopher traps, kind of the mechanical ones that go underground. This could be blocking pests out or making the environment unsuitable for it. So, you know, putting bird tape up in a farm to like the metallic kind of blowing in the wind bird tape, or the bird eyes to try to keep birds out, mulching for weed management, barriers, like physical barriers to keep birds and insects out. The photo is of a sticky trap, so you're able to kind of see the insects that are flying around. So yeah, also another mechanical one is spraying plants to walk with water to knock off aphids. So after we try some of these and none of them worked, we can move on to the third step in the pyramid, which is biological controls. So again, this is kind of the third step because it does introduce some new species to your farm, which is not something we typically want to just do lightly. So in biological control, we use the natural enemies of a pest to control them and their damage. So this includes introducing predators, parasites, pathogens, and competitors. So some examples of this are growing insectary plants such as yarrow that would attract predatory or parasitoid insects that prey upon your pest insect. Another insect that could be a good predator to introduce is a mealybug destroyer if you're having a mealybug infestation, because this would control a pest, but you are again introducing a natural enemy of the species, but it's of a foreign origin. So we're not running to this step first. We want to try to use the first two parts of the pyramid before we introduce these new species. So if we tried, again, all of that and it didn't work, the top of the pyramid is chemical control. So that's using pesticides, herbicides, and fungicides. The photo on the right is an example of a rodent bait station that includes chemical bait for rodents such as gophers, ground squirrels, and this is a more aggressive form of like a mechanical gopher trap. So when we're using chemical controls, there are some considerations we should take. One of them is pest identification, so accurately identifying the pest to select the appropriate pesticide. Monitoring the pest, so we're really, observant and we're really aware of what is going on, so that we're making the best decision for the farm, the pest, the crop, and the environment. Safety of humans handling its storage and application to minimize overall risk. And then resistance management, as John mentioned, we are using chemical controls as a last resort, because if we turn to them all the time, we can build resistance in the pest populations, and that would be detrimental for long-term effectiveness of chemical controls in certain crops. So these are some resources for IPM assistance, and do you want to say anything about the resources?
I do want to mention something prior to what you were saying. I did want to mention that you could use your cultural, physical, and biological controls at the same time. You just have to be wary about to if you're going to use biological controls, you want to make sure that you're not obviously harming the pests that you're currently introducing to protect from the bad pests. The only one that you can't use interchangeably with those is going to be chemical. Chemical is just going to take everything out at that point. That's all I wanted to mention.
Okay. So yeah, the resources for IPM assistance, we have the UC IPM program with UCANR, your local RCDs. The Cornell IPM website has some great resources. Obviously, the Santa Barbara Botanic Garden is a wealth of knowledge, as is the CEC, and then contacting a CCA or a PCA. And then if you scan this QR code on the right, there's just a list of resources that we have compiled of IPM, related things. Now that we talked about the controls, I would love to learn about what you all are implementing on your farms, vineyards, etc. So does anyone have cultural controls that they that work for them, or they heard of, or that they just already do, on the daily?
Feel free to unmute yourselves if you have an idea or suggestion. I will say, Stacy, you.
Yes, so this is Stacy. One of the things that we've battled are ants. Pastoral I firmly believe Pastoral is built on a giant ant hill. And so with that, we get mealybug pressure because the ants are moving them around. So this year, we've laid out organic bait in the past, and it's relatively effective, but in hot spots, it's hard to control. So this year we decided to use water crystals and boric acid, and which was a study that came out of UC Davis in the last couple of years. And so we're finding that extremely effective because the ants will come in because they're thirsty, take that water crystal back to the nest. And so for me, that's been something that's been surprisingly effective.
Unmuted.
We've have a really ants too. Thank you for that suggestion, because we're dealing with it right now as well. Like no matter what we do, we just can't seem to get rid of them. And we tried organic bait and it just doesn't seem to have helped us at all. Also our squirrel problem is just out of control. We're doing the traps, trying to do it a more organic way, but it's just hard to keep up with them. So if anyone has any other strong suggestions on how to get rid of that squirrel or get that squirrel population down, we'd appreciate it.
Carbon monoxide.
Carbon monoxide. Yeah, I've heard that. We haven't tried that yet.
Very effective.
Okay.
For cultural controls, we have farmers using solarization for weed management.
We've also heard at other trainings we've held with the carbon monoxide for squirrels and other rodents. Some soil types aren't as conducive to, like, if you're putting the smoke bomb in the hole, being able to cover every single hole, because some soil, like cracking, allows for the smoke to kind of get out all over the place. So yeah, people are saying they're having.
We've tried that, using those, and these guys are so smart. They throw it back out at you, basically. Yeah, we just stand there scratching our head. So we've had trying to cover holes, but there's just so many. And then you turn around and you see it laying out where they just pushed it right back out the hole. So
there is another device called a Rodinator. You do need a trained professional to use it, but it's a device that combines a mixture of propane and oxygen, and you insert the device into the squirrel holes, and then you let it go for like 30 seconds, and you light it, and it creates an explosion inside the holes. It ends up killing a bunch of the squirrels, and they can't smell it, so a lot of them get surprised, and it's very effective.
Thanks. Yeah, I know that Lake Casitas had used that several years back, and you look like the whole earth just exploded when they did it. But yeah, we've been told by some of our friends who are on the board for the Casitas Lake that was a suggestion, is trying to look into that. So thank you.
Oh, sorry.
Regarding, like gophers, we had several farmers really liked using the gonzo traps. No, gonzo, the brand. That's more of a physical thing, but
And I don't remember the details of this, but wasn't there talk about planting like a sacrificial crop at our
Yeah,
last?
There is that's for gophers though.
oh,
There is a plant called the gopher plant. It's a type of euphorbia. It's a bit invasive, but it has a latex root system that prevents gophers and sometimes squirrels from actually being in that area. Like I said, the only main issue to it is it's an invasive plant. It does throw out a lot of seeds, and then you'll start finding it throughout your orchard. But once the gophers or squirrels do bite into those root systems, it can kill them, or it'll just really highly deter them away from that area.
I started using that in my garden. It's a very large garden, and you are absolutely right. It's invasive. It just comes up everywhere within the garden, and even in my boxes, which I'm like, how to get up there. But and then it's starting to show around the garden area. So, it's constant weeding and pulling them out when they're young, and making sure you want it exactly where you want it, because it really does start trying to take over.
The one thing I will say about the gopher plant, it's very effective if you're able to cut off the seed stalks
oh,
before they actually seed. And it's a very big, beautiful flower, but if you can cut that off and find before it seeds, you control it very well that day.
So some of these were not technically cultural, like, the traps would be physical and mechanical, and like planting this gopher plant would technically be biological, but I will sort them later. But yeah, if no one has more cultural things, let's talk more about physical mechanical controls. So more like tangible physical. I know some were already mentioned. People using any other traps or
Owl boxes, I hope I'm not stating the obvious, but owl boxes are really effective.
Yeah, I was going to say that in our predator purchase too. We've started putting a few more in different areas to try to help.
yeah, even if you feel like something is like obvious, I would still say you should say it aloud, because you're all in different regions and farming microclimates and dealing with pests differently, so it could be helpful for someone else.
I'll second the owl boxes. We have several on the ranch, and they definitely do make a difference. Same with, I was looking up the gonzo traps. I didn't know that's what they were called. I've heard different names from, but for trapping gophers, they're like the square metal frame with the prongs. We've used those for the last 10 years on the vineyards we farm. And the thing with those is it's just consistency. And using them early in the morning when you can see the fresh gopher holes in the fresh soil, feels like the fresher the mound, the easier they are to catch. And then squirrels are like the bane of my existence. And so we spend a lot of time trapping squirrels, we use the squirrelinators, the metal cages, and we find that chicken scratch from the coop is like, candy to them. And so we use that as the bait to lure them. And then again, just like trapping gophers, trapping squirrels, it's just about consistency. And it's not about total elimination, but it's just about like controlling the population at the end of the day. Yeah.
The one thing I will say about using a squirrelinator, the laws on using that one, just because it is a live trap, they vary between counties. So I'd say check on that first before you use a squirrelinator.
Yeah, thank you for mentioning that.
And I guess some other examples that other farmers have indicated to us in previous, previous times, they've mentioned like using bird lasers on vineyards, using flash tape also.
For bird control, we've used it on our property, but my contract growers often use falconers.
Oh,
Yeah, I can second the bird lasers. I probably have a dozen of those throughout Santa Barbara and San Luis County, and they work pretty well along with the flashing tape.
Cool. This goes back to cultural controls, but shifting our perspective on like what is a weed, and like maybe there's actually a benefit to what we typically consider a weed. Maybe it's breaking up the hard pan in the soil, or it's attracting pollinators, or allowing for better water infiltration or moisture retention in the soil. So about the flame weed. Yeah.
there's a flame weeder, you can use that propane tank attached to basically, I don't know what's it called, a torch.
Flamethrower.
Yeah, it's basically a flamethrower to kill weeds. But typically for that, you have to get the weeds when they're young, at a young age.
Does anyone use a flame weeding? Did anyone say they use a falconer, or just people in the area use that?
In the past, I've used it on owned vineyards, but I have a lot of contract vineyards in Paso Robles that use them.
Cool. That's awesome. Okay, so physical, mechanical, that's some great examples. I know rodents, squirrels, gophers are always, always the main conversation. I feel like when we're talking about physical and mechanical controls, and it seems like we hear that a lot of times people are just setting, 30 to 50 plus traps and checking them every day. But it's good always to hear more examples and more ideas. Okay, so biological controls, we talked about planting gopher plant. Does anyone use any insects or any other plants that are being used as more of an insectary or like control?
So I planted a 3/4 acre vineyard block with alyssum and marigolds and poppies and monkey flower, but specifically I planted the alyssum for a host for lacewings because they help control the mealybug population, and they eat a lot of bed bugs. I planted the marigolds for some level of gopher control because they're resinous and they'll be more resistant to gophers. And surprisingly in that block, I have gophers everywhere in my 40-acre vineyard, but that 3/4 acre vineyard doesn't have any gophers.
Cool.
Did we talk about pheromone control at all or pheromone trapping? I wasn't sure if that was mechanical or behavioral or biological, excuse me.
That's gonna be physical.
Physical.
Under physical, okay.
Yeah, some of them kind of feel like they span, you know, different ones.
It can even enter chemical.
Yeah, it could even technically be.
Yeah.
chemical.
We've had good luck with pheromone control for, or pheromone trapping for mealybug.
Sorry, my name is Geronimo. I was having problems with the microphone, but I've been using flares with great results. I have 10 acres of pastures, and I've been also using photoxin, which is some pills that you drop in and they create a gas. You need to cover the hole, same as the flares. And those two have been working very well.
Layers and what was the other thing you said?
It's called photoxin.
Boat.
It's aluminum phosphate, I believe.
Aluminum phosphate?
I think so. I'm gonna
Okay,
check on
sorry.
the. Yeah.
I was having an issue hearing. Cool. Sorry.
I would second the flare use, especially for squirrels. But you just have to be careful because you have an open flame, and especially, I know on the Central Coast, they'll invade, and set up colonies around oak trees. So you just have to be cognizant of the roots and all that. But, flares work tremendously well.
This wouldn't technically be biological. I think it would be more physical and mechanical. Yeah.
Going back to biological, does anyone use different livestock or animals to address pest issues?
This year I utilized, 2 different teams of goats and sheep for weed management. One, for some vineyard weed management before, bud break, and then to prepare for planting in our Dry Creek bed, for this grant. I had a big team of goats and sheep on the property for about 6 weeks.
Awesome. Great.
Ladybugs?
Does anyone put any insects onto their farm, like ladybugs or anything?
We've used ladybugs and that, also natural predators for our problem with the squirrels. We've got several bobcats that live on our property, because we're surrounded by so much open space with our ranches that surround us. So we're lucky with that we've got, those natural predators that actually help, you know?
Gopher snakes are another natural predator.
Yeah, we love them. California kings and gophers, right?
Great, that's an awesome list. Moving on to chemical controls, any herbicides, pesticides, fungicides that anyone uses that you know works for them? So you have the yes, bait trap. For rodents. Does anyone use the T bait trap for rodents? No. People have mentioned. Habitat.
At the RCD, our restoration team regularly uses a vinegar, salt, and soap mix. And it's organic. And it can kill even,
Nutsedge.
nutsedge.
It's good at killing nutsedge.
There's also, people have mentioned like essential oils that they use,
Neem oil.
yeah. neem. Um,
I mean, there's around.
If you were not organic, Roundup.
There's a bunch of chemicals they can use, so.
Yeah, I think the goal of the PHP program is we're bringing in all of these pollinators and we want them to stay there. We want them to survive. So if we can be conscientious about, you know, when, where, like how we're addressing the pest issues. So if you aren't organic and you do spray, yeah, really paying attention to those labels so that we're trying to keep the beneficials around as best as possible.
Thank you. Okay. So, yeah, now we're gonna pass off to Sarah from the Botanic Garden, who's gonna talk more about monitoring and pollinators.
Thank you, Christina. Nice to meet you guys all. Again, my name is Sarah and I'm from the Santa Barbara Botanic Garden. I'm one of the ecologists. And you guys did a great job of talking about all the creatures we're trying to get rid of. I'm going to focus on the creatures that we want to try and bring in. So all of our beneficial insects and pollinators. And I will share my screen real quick. Can you see my presentation? Yeah, okay. Before I get too far into this, I just wanted to say that a PDF version of this presentation that has more text on it, so it has all of the text that I'm saying basically, will be available as well as both forms that I'm going to talk about, because there are two monitoring forms, monitoring form 1 and monitoring form 2, and those will be available as PDFs. And then lastly, there will be a kind of course pollinator identifier guide that has lots of pictures and diagrams. And so that will also be made available. So everything you see here, you will have a paper version of too. Just wanted to say that before I got too far. And so I also wanted to talk about what is a pollinator and what is pollination. And so this might be pretty redundant to a lot of you, but just in case. A pollinator is anything that helps carry pollen from the male part of the flower to the female part of the flower. And so this can be a bird, a bug, a dog, a person, anything that's moving that pollen between reproductive parts. About 80% of the world's flowering plants benefit from pollination. So of our wild plants, of our cultivated plants, about 4 out of 5 need something to move that pollen for them between the male and female parts. And that translates to about 1/3 of our food crops and 2/3 of the food crops that I would say that we especially like. So 2/3 of the foods that are fruits and vegetables and nuts and seeds and all of the especially delicious stuff is brought to us by pollinators. And so all of these depend on animal pollinators for their reproduction. But one thing that I always like to highlight is that pollinators are not necessarily doing this altruistically. It's not like they wake up in the morning and plan to go pollinating. They're visiting in these flowers because they're in search of food, which could be the pollen itself, shelter, nectar, and sometimes they're even looking for mates. But the plant, it's lucky for the plant that the pollinators are just super sloppy eaters. So they just get the pollen all over themselves while they're doing this foraging and then accidentally move that pollen between plants. And so the goal of the PHP program overall, which was nicely stated earlier, was just to establish and document resources for a broad range of pollinators. And 3 things that I wanted to note as we talk about the monitoring. When you guys accepted this opportunity to place habitat on your properties, you are required to do 3 years of monitoring following that implementation. So 3 years following putting that practice onto your landscape, you're required to monitor. And we've tried to make the monitoring as easy and straightforward as possible, but I did want to just stress that you are required to do it. And I do see a chat. Oh, and that was from Christina. Okay. Monitoring should occur 1 time per year. This can happen in the spring or the fall or whenever is most convenient for you, but we do need it to happen 1 time per year. And you are responsible for undertaking and submitting this data to the CEC in a timely manner. And we'll talk about how to best submit this information at the very end of the presentation. So those are just the notes. You're required to do it for 3 years, 1 time per year, and it's on you to submit that paperwork. So we're going to do 2 methods when we are monitoring to see how effective our habitat is. The first is we're going to take photos. So photo points are really specifically to assess the survivorship and the success rate of the newly installed habitat. And we'll go over how to take those. And then the next method we're going to use is a transect monitoring. And this is really more about measuring the pollinator abundance and diversity in the newly established habitat. So the photos are really to document that you put the plants in, and then we're going to talk about transect monitoring to make sure that those plants are providing the resources we wanted them to pollinators. So the photo points. For each PHP practice you put out, so whether this is a hedgerow or a conservation cover or riparian habitat, you need to take 3 to 5 photos for each practice. Each photo should be marked, numbered, and uniquely identified, and we'll talk about how to do that. And if you have a hedgerow in multiple places or a practice in multiple places, each practice has to be photo monitored separately. So like I said, if you have a hedgerow on the east side of your farm and one on the west side of the farm, those need to be photo monitored separately. And so how does the photo monitoring work? So this is one of the forms that will be available as a PDF. And this on the left-hand side is a hypothetical image of your marked photo point. So you guys can mark it in any way that is convenient to you. You want to be able to take the same photos each year. So ideally you would be able to semi-permanently mark it either with a stake or a tag, or some sort of geographic reference. So if you have a tree that you can put your back up against and always be taking in the same direction, that is great. So let's say this is actually a picture of the Santa Barbara Botanic Garden meadow, but let's say that it is a conservation cover that I put in. And I've marked my plot that I'm going to take 3 to 5 photos of this per year. And so when I'm filling out the form, I'm going to go ahead and record the date that I did it, the farm that I'm a part of, any notes that I have about the weather or something interesting happening. So on May 1st, there were a lot of butterflies around. Some way to identify this photo, and this is just on my camera. It was the 635th photo taken. What exactly the PHP cover or PHP practice is that you're documenting. So this is a hypothetical conservation cover. And you want to record the GPS point, and we'll talk about the best way to go about this also at the end. So you can either do this on your phone, or there are a couple of different types of apps that will allow you to actually physically paste the GPS point directly onto your photo, which is really what we recommend, because then your photo, we always know exactly where it was taken. The direction of the photo, you want to be taking your photo in the same direction each year. And then any descriptive notes that might help you in the following year. So this is a photo of the conservation cover with a meadow and a mountain in the background. And so for each PHP practice, you're going to be taking 3 to 5 photos of different angles of the same practice every year for 3 years. And that is primarily to understand how the plants are doing that you've installed as part of this PHP practice. The second part of the monitoring is the transect monitoring. And this, you'll only set up 1, possibly 2 transects for your entire site. So this is really independent of how many different types of practices you put onto your site. You're going to set up a centrally located 100-foot transect that you're going to slowly walk for a 10-minute observation period. And you can have 2 of these if your site is especially large, or if it's broken up by a large amount of land, you can put one at 2 locations. And so the way the transects work is you're going to lay out 100-foot tape or measure in some way. If you know how long your paces are, you can pace out 100 feet. You can measure it once and mark it. And then you're going to very slowly walk 10 minutes down that transect, noting pollinators along the transect. And ideally, your transects will be located in the same place each year. So this is kind of a diagram of what a transect could look like. So we have our GPS at the start coordinate, again, marked so that we can do the same transect each year. And then you'll walk very slowly down 100 feet in a 10-minute period. And it's an amazingly slow walk. When we've had this training in person, we've been able to practice it. And it's pretty startling how slowly you actually have to go to be able to only walk 100 feet in 10 minutes. And as you're walking, you're going to be wanting to look 5 feet in front of you, 5 feet to either side, and 5 feet above you. And within that kind of bubble, you're going to note down every pollinator you see. And I'm going to talk about what is a pollinator specifically and how to identify it in just a second. But this is kind of the setup of the transect. And again, you're going to want to place this somewhere on your property that is centrally located, that you think would be benefiting from the practices you've put out. So if you put a hedgerow, it would be a perfect opportunity to do a transect down that hedgerow. If you have a big patch of cover crop, maybe directly through that cover crop, something like that. And this is definitely, locating your transect is the most site-specific and tricky thing. And this is definitely something that I'd be excited to work with folks individually if you wanted to chat and figure out where to put your transect, you know, just to best monitor the bugs that you're getting. And then a real quick reminder of what a pollinator is. So you guys remember earlier, I mentioned that it's any creature that moves pollen from the male part to the female part. And so an important part of that is the insect has to be touching the reproductive parts. So if I'm walking and within 5 feet of me, I see this beautiful Grandilia flower, this insect here, this long-horned bee or Eucera, would be considered a pollinator because it's actually touching the reproductive parts of the flower. Whereas this little tiny Halictus, in this moment in time, would not be considered a pollinator because it's on the petals of the flower, or if it was on the leaf or a branch or somewhere flying nearby. In this moment, I wouldn't consider this a pollinator on my walk. And I'm happy to talk more about that distinction if it would be helpful. But just again, they have to be touching the reproductive parts of the flower. So in this Grandilia, it's the center part, and this Halictus is not doing that. So when you're recording, when you're on your transect and you're very slowly walking in a meditative way, we don't expect you to be able to identify every pollinator to species. And so it's commonly agreed upon that there are kind of 7 groups that we ask folks to clump their pollinators into. And this was decided by folks at Berkeley and the folks at Xerxes. And it's kind of a way that all of our data can be comparable, and we're kind of all doing it to the same level of specification. And so there are 7 groups that we're going to kind of coarsely try and lump our different pollinators into. And I'll talk about each and how to identify each of those. So the first 3 groups are, I would argue, the trickiest to tell apart from each other. The first group are our European honeybees, and we'll talk more about them in a second. The second group is a group of solitary bees, which sometimes can look pretty similar, but are very ecologically different. And our third group are the bumblebees, also native to this part of the world. Our fourth group will be the wasps. Our fifth group will be the butterflies and moths. They all get lumped together. And then our last 2 groups are the flies and the beetles. And we're going to talk about how to identify each of these. But again, this is just so everyone's kind of recording information in the same way, given differences in our ability to identify bugs as they're flying by at 10 miles an hour. So the honeybee, Apis mellifera. This is a bee that it sounded like a couple of people are keeping on their properties. But this little critter has been domesticated and can maintain feral populations around, and is probably the most common pollinator that you'll be seeing. I did want to stress that they're not technically native to North America, so they fall into a different group than our native bees, but they are important pollinators. And the best way to tell these guys apart, they are the only bee with a hairy eye. And I know that's really tough to see when they're flying quickly, but it is a surefire diagnostic characteristic. And another really important thing about the honeybee is that they have flat hind legs. So you see this sticky ball of pollen. This is being collected on what's called the corbicula. And it's this kind of naked part of the flat back leg that have long hairs that make a basket that the bee will put in wet pollen to carry around. So if you ever see like a big glob of wet pollen on a back leg, that's probably a honeybee. And I wanted to stress that honeybees can vary a lot in color. So you can have these really blonde ones and these really malefic darker ones, but these are all honeybees. And once you get a knack for it, you'll notice like you can tell a honeybee apart pretty quickly, especially always looking for those flat back legs. Okay, our next group is way more diverse. So rather than that one species of honeybee, we have roughly 1,600 species of solitary bees in California, and they vary widely in size, in shape, in color, in behavior, and all sorts of things. The one thing that groups them all together is as solitary bees, they don't live in a hive, they don't live in a colony. They make solitary nests. The females will provision their young with the pollen they collect individually. And so they're not making honey or anything like that, but they are very effective pollinators. And again, they're native to this part of the world. And we have some of the, I would argue, the best native bees in the US. But so there are different groups that I'm just going to kind of touch upon. And again, we have a nice PDF handout that you guys can take into the field with these big pictures and kind of help identify the different groups. But some of the solitary bees, they'll collect their pollen on their tummies rather than on their legs like we saw in the honeybee. But they've got these very fluffy tummies that you can kind of see here, and they'll be covered with pollen. And they can be an iridescent blue or green if it's an osmia. They can have black bodies if it's a megachile, but they'll both have that hairy abdomen. They can be very, very tiny, like these little sweat bees or lasioglossum, and have a dull iridescent body. And they can have, the solitary bees will have hairy legs rather than that naked flat leg of the honeybee. And then we have these much larger, more charismatic bees, including this densely furred digger bee, or the xylocopa, which is a carpenter bee, which would be very large. And so I'm just really trying to impress upon you the variation in solitary bees. So like the honeybees will all look kind of similar to each other. And then we've got this wide suite of very dynamic, different solitary bees. So these are more solitary bees. Some solitary bees can have really long antennae, the longhorned bee. They can have little stout abdomens. They can have long antennae and little noses that are yellow. They can have bright green bodies, like little race cars. So really diverse and dynamic, this group. Our next group, our third group, are the bumblebees, and they're about 25 species native to California. And these are pretty easy to tell apart. They have really densely furry bodies and usually slightly darker wings. They're much larger and chunkier than a solitary bee for the most part, and a lot of times you can hear them coming. They have like kind of a deep rumble in their flight. And they actually are the closest related to the honeybee that are native to North America, and they have similar flat back legs that they use to collect this wet pollen blob. So they're similar to honeybees in that respect. Large fluffy bodies. Our next group are wasps. So all bees are vegetarian, and they feed their young bits of plants that they've collected, so the pollen and nectar that they've collected. And the big difference with the wasps is that wasps are carnivores. And instead of feeding pollen to their young, they feed meat. They feed small other insects that they've collected. They feed little pieces of hamburger if they can get away with it, anything that they can find. So one important distinction is they do not have fluffy bodies for collecting that pollen or that plant material. They have more of like a predator-shaped body for collecting meat and carrion. So that's a big distinguishing characteristic. They're just generally hairless. They also tend to have these long, thin waists, one of the wasp waist characteristics. Sometimes they'll have folded wings. So these wings will actually expand when they're flying, but when they're at rest, they'll fold them into this kind of sleek pattern. They can have really long ovipositors. And these are parasites and parasitoids that actually lay their eggs in other insects. So they'll have these really long looking stings. They aren't dangerous, but they're just for being able to perfectly position an egg. And then it can also be super tiny. So this one, the parasitic wasp can be less than 1 millimeter long. But generally, they have thin waists and sleek hairless bodies. Our next group are the butterflies and moths. So there are nearly 5,000 species of these in California. And the most important groups are the skippers, the butterflies, and the moths. So the skippers have little hooked antennae. They're kind of hard to see here, but you can kind of see them here. And when they're at rest, they have their wings folded in this really distinct pattern. And they tend to have kind of a rapid, haphazard skipper flight pattern. So between the hooked antennae, the wings folded at rest, and then a very distinctive flight pattern, you can tell that it's a skipper. Butterflies only have clubbed antennae. So this comes to a little club here at the end rather than a hook with the skippers. And I'm sure people are pretty familiar with butterflies. They can be super diverse, super beautiful, wide range of colors and shapes. Sometimes they can have these little tail-like projections on the bottom, like with this hair streak. And then the moths, their night-flying cousins, are actually even more diverse. And the best way to tell them apart is usually they're flying at night, but there are some day-flying moths that you'll see, but they will always have very plumed antennae. They can be very diverse, they can be fluffy, they can be straight. So really the best way to tell apart a skipper from a butterfly to a moth is the type of antennae they have. Skippers will be clubbed, skippers will be hooked, butterflies will be clubbed, and then moths will have a diverse set of different types of antennae, some of them plumed. They tend to have thicker, fuzzier, heavier bodies. Good way to tell them apart. And then on the bottom, I just wanted to highlight, these are all about 1 millimeter long, and most of the diversity of butterflies and moths we have are these tiny microlepidoptera, which are very difficult to tell apart, but important pollinators in many of our species. Our next group are the flies. So flies are in the order Diptera. Diptera means 2 wings. And so the biggest difference between flies and our other pollinators is they only have 1 set of wings. So there's 1 set here. And then their second set of wings that the other ones have are reduced to these little lollipop structures called halteres. And so all flies only have 2 wings, a single set of 2 wings. These are these mimics that mimic honeybees, and so they're trying to look tough, but they're not. These are all hoverflies. And you can tell they're hoverflies first because they only have 1 set of wings, and also they have giant eyes. So a good way to tell a fly apart is how giant its eyes are. They take up most of the head. And they also have a very characteristic way that they fly. They'll hover, which is why they're called a hoverfly in many situations. And then other types of flies, they can have these stiff bristles. They can have very, very short antennae on their face. They look like little mustaches sometimes. Can have red-tipped abdomens. They can also be very iridescent, like some bees, but they'll tend to be like covered in these very stiff bristles. And then our last group are the beetles, which are about 8,000 strong here in California. And the best way to tell them apart is they have 2 sets of wings, but one of their sets of wings is hardened into something called an elytra, and that keeps them protected. And so this can be scaly or hairy or shiny, but all beetles will have a hardened elytra or that hardened front wing set. They have super variable antennae. Some of them can be clubbed, some of them can be filiform. These are the hardened front wings again. Okay. So real quick, if people are willing to participate, I'm going to put a picture of a bug on the screen. And if folks wouldn't mind calling out what they think it is and why. Anybody have any idea what this guy is and why?
Honeybee, hairy eyes.
Yeah, honeybee, hairy eyes, and flat back legs. Great, thank you. Honeybee. What about this one?
Okay, I know fly, right? But I'm not sure the name, sorry.
This one's actually a wasp. So I can see where you're coming from. It has huge eyes.
Looking at the eyes.
Yeah, no, and that's really good. But it has this really long waist. And in theory, you'd be able to tell that it has 2 sets of wings. So 4. But that's a really good guess. This one's actually a wasp. What about this one? I think Ventura RCD, you guys are allowed to guess too.
Oh, it's a fly.
It's a fly. Yeah, so this is one of the hover flies that's mimicking a bee. It has 1 set of wings and huge eyes, very short antennae right there, and a little bit of hair. So yeah, this is one of the syrphid flies. What about this one? Any guesses? Bright iridescent green, very fuzzy, 2 sets of wings, long antennae, hairy legs.
A Hymenoptera?
It is a native bee, yeah, which is a Hymenoptera. Yeah, so the other Hymenoptera that would have been included would have been a bumblebee. So this is not a bumblebee, or a honeybee, which this is not a honeybee, or the wasps also fall in that order. And this is not a wasp because it has kind of a thick waist and it's very hairy. But yeah, it's definitely a Hymenoptera and a cute one. Solitary bee. What about this one? Narrow waist, folded wings, very sleek, hairless body, long antennae.
This one's a wasp.
Yes,
The
thank
long,
you, Gina.
yeah, you got it.
But sorry, go on. Why? Why did you think it was a
Oh,
wasp?
the same reasons that you had mentioned, but the long, thin waist, the folded wings,
Mhm.
or they're not folded in the photo, but you know what I
They're
mean.
more they're more folded than they would actually be when they're flying. They kind of are twice that width when they're actually flying.
Great.
So thank you, Gina. Wasp. What about this one? This one's a little tricky. Any ideas?
Is this a solitary bee? Because
This
he's carrying
is.
pollen all over his legs.
Yes. So this is a solitary bee with very hairy legs. She's got naked eyes, 2 sets of wings. Yeah, and I think the best diagnostic characteristic is this very hairy leg, just covered in pollen. So great spot. What about this one? Got a big chunky body. You could probably hear her coming.
Bumblebee.
Bumblebee, yeah. So this one would be a bumblebee. Like I was saying, a very densely hairy body, black and yellow. She would also have a flat back leg, but you can't see her carrying pollen right now. And what about this one? This is the last one and probably the trickiest. This one is actually a honeybee. And And I put this in here because it's so dark, it might trick you into thinking that it's a solitary bee, but she actually has very hairy eyes. And then you see this big, flat back leg with curved hairs that would be carrying her wet pollen. So this one is a honeybee. Okay, so how are we recording these things on our transects? We set up our 100-meter tape, we start our timer, and then we start walking. The things that we need to do are record the date, the time that we started our transect. We really want to be doing these transects during ideal weather, so like pretty sunny, not too windy, not too hot, if possible. We can number our transects, record where we are, record the GPS of where we started, and the direction, any comments you have about the weather around you. And then as you're walking, you'll just tally the number of insects you see. So in this hypothetical, I walked for my 10 minutes and I saw 4 honeybees visiting flowers. I saw 2 native solitary bees. And then there's another category. And so this will be anything that either you cannot identify or anything that's not listed here. So if you saw a spider on a flower, if you saw a hummingbird, if you saw something that you just didn't know, and then you'll total those up at the bottom, and that's what you'll be submitting to the CEC. And so at the end of both the photo monitoring and the transect monitoring, you'll have one sheet that is the Form 1 that has all the details of the photos you've taken, along with all the photos, and then a second sheet of paper that will be Form 2 that you've just recorded what you saw on your very slow walk of your transect. And the CEC will have a folder dedicated to your farm and project in particular that you'll upload all of that data. And so all the photos and all of the data sheets will be submitted once a year and stored in this folder for submission. So just a quick, how much should I survey question? Say hypothetically you put in a single acre of hedgerow, or sorry, you put in 1 conservation cover in your area and 1 hedgerow. What are you required to monitor? So remember, for each PHP practice you put out, you have to take 3 to 5 photos of that. So you'll take 3 to 5 photos of each conservation cover area. So that's 3 photos. 3 photos of your hedgerow. So 3 photos, and then only 1 pollinator transect for your whole farm that year. What if in a different situation I had 2 windbreaks, 1 hedgerow, 2 cover crops? What do I need to submit to the Google Drive? So again, you need to have photos of each of your PHP practices. So 3 photos of each windbreak, and because there are 2 windbreaks, 6 photos. 3 photos of each hedgerow, and there's 1 hedgerow, so just 3 photos. And then 3 photos of each cover crop, and there are 2 cover crops, so 6 photos. And then a single pollinator transect for that year. And I'd be more than happy to talk through exactly what PHP practices you're putting out on your property and exactly how to make sure that you're getting all of the monitoring done that you need to. And if we were in person, we would go practice this as a group and recording, but we are not. But just for some more resources, and this will also be on the PDFs that you're sent. All of our monitoring protocol is based on the streamlined Xerxes protocol, which is discussed in detail. And then if you need help with either plant or pollinator ID, iNaturalist is a really great resource, as is Bug Guide. And then this GPS map camera app is an app you can put on your phone, and it will directly apply GPS points to your photos. So like on the photo itself, it'll read the GPS point, which can be really convenient because then you don't have to worry about recording that as well. And with that, that is the end of my presentation, and I'd love to answer any questions. I'm sorry we can't be in person and go look for bugs together, because that is the best part.